DNA sequencing chemistries. Cycle sequencing is a simple method in which successive rounds of denaturation, annealing and extension in a thermal cycle result in linear amplification of extension products. These products are then injected into a capillary. The cycle sequencing is focused on two general categories of sequencing chemistry approaches: dye primer chemistry and dye terminator chemistry. Sequencing with dye primers. 4 separate reactions are performed when using dye primer based sequencing chemistries. Each reaction is labeled at its 5' end with any one of the 4 different fluorescent dyes. Each of the 4 sequencing reactions will contain fluorescently labeled either a blue, green, yellow or red labeled primer. The color of each reaction corresponds to either A, C, G or T. Dideoxyribonucleotides (DDNTPs) are present in each reaction mix and randomly terminate DNA synthesis, creating DNA fragments of varying length. Since a fluorescently labeled primer is used for extension, all terminated fragments are fluorescently labeled. Following a sufficient number of cycles to allow for optimal generation of extended products, the 4 reactions are combined and electrophoresed. Sequencing with dye terminators. Fluorescent DNA sequencing can also be performed using a chemistry in which the dyes are attached to the DDNTPs, thereby requiring only 1 reaction tube per sample instead of 4. Since only 1 reaction tube is required for the dye terminator reaction, this chemistry is simpler to use than dye primer chemistry. DNA template, unlabeled primer, buffer, the 4 DNTPs, the 4 fluorescently labeled DDNTPs and DNA polymerase are added to the reaction tube. Fluorescent fragments are generated by incorporation of dye labeled DDNTPs. Each different DDNTP, that means the DDATP, DDCTP, DDGTP or DDTTP will carry a different color of dye. All terminated fragments, those ending with a DDNTP, therefore contain a dye at their 3' end. Future methods or next generation sequencing, NGS technology. Next generation or second generation sequencing technologies have largely supplanted first generation technologies. These newer approaches enable many DNA fragments, sometimes on the order of millions of fragments, to be sequenced at one time and are more cost efficient and much faster than first generation technologies. The utility of next generation technologies was improved significantly by advances in bioinformatics that allowed for increased data storage and facilitated the analysis and manipulation of very large data sets, often in the gigabase range. The RNA sequence assay, relying on the next generation sequencing instruments, provides more comprehensive information on a sample's RNA contents, known as transcriptome data. Another noteworthy example is the ChIP-seq assay, which combines next generation sequencing with ChIP or chromatin immunoprecipitation for detecting the genome-wide DNA binding sites of a protein of interest. Noise of these data comes from possible sequencing or base calling errors and the need to map the sequence segments back to the genome. Several methods have been developed to analyze these data using ad hoc approaches or some probabilistic modeling. Applications of DNA sequencing technologies. Knowledge of the sequence of a DNA segment has many uses. First, it can be used to find genes, segments of DNA that code for a specific protein or phenotype. If a region of DNA has been sequenced, it can be screened for characteristic features of genes. For example, open reading frames or ORFs, long sequences that begin with a start codon, that means the 3 adjacent nucleotides, the sequence of a codon dictates amino acid production and are uninterrupted by stop codons which, except for one at their termination, suggest a protein coding region. Also, human genes are generally adjacent to so-called CPG island clusters of cytosine and guanine, two of the nucleotides that make up DNA. If a gene with a non-phenotype such as a disease gene in human is known to be in the chromosomal region sequenced, then unassigned genes in the region will become candidates for that function. Second, homologous DNA sequences of different organisms can be compared in order to plot evolutionary relationships both within and between species. Third, a gene sequence can be screened for functional regions. In order to determine the function of a gene, various domains can be identified that are common to proteins of similar function. For example, certain amino acid sequences within a gene are always found in protein that span a cell membrane. Such amino acid stretches are called transmembrane domains. If a transmembrane domain is found in a gene of unknown function, it suggests that the encoded protein is located in the cellular membrane. Other domains characterize DNA binding proteins. Several public databases of DNA sequences are available for analysis by any interested individual. Sequencing technologies also play a key role in the assays that require the identification of DNA sequences. Examples of such use include the serial analysis of gene expression and its updated version, the CAP analysis of gene expression. Both methods measure the relative frequency of different RNA transcripts in a high throughput fashion. The applications of next generation sequencing technologies are vast owing to their relatively low cost and large scale high throughput capacity. Using these technologies, scientists have been able to rapidly sequence entire genomes, that means the whole genome sequencing of organisms, to discover genes involved in disease and to better understand genomic structure and diversity among species generally.